Welcome to Study Time, a televised home learning program produced by Rwanda Education Board.
Here we are again. Welcome viewers back home to these learning sessions for TV series for you, the candidates, and other students back home. Remember, like we said, this does not only concern students who are going through their learning processes at home because of these businesses going on, but as well anyone who is at home and is able to take advantage of this should sit, relax, and follow and enjoy the sessions. So today, to the S5s, and also a reminder to the senior sixes that we are looking at Unit 6, protein synthesis, making of the proteins by the body, and we are also looking at exactly what is made, but also how. What is the very beginning of all this? So in today's lesson 1 of the first unit, 6, lesson 1 of unit 6 for protein synthesis, we are looking at only just one objective. Now, there is something I really need to take you through here so that you always remember. In some way, this is some kind of rather abstract topic in which us as learners tend to only pick up the information and we've never been able to get inside our body and know exactly what's going on. So in most cases, us as learners tend to pick the information and just say, you know what, let me grasp it after it is going to come in the exam, and that's it. But is there a component that we somehow lose out? So let's get back to a few small things before we move on. We talked about the DNA, we talked about the different forms of RNA in the lessons previously, in which we were looking at the chromosome, then the components, then the DNA, RNA, the differences between the two, and other forms of RNA. And I remember mentioning, if you recall, that these other forms of RNAs can be looked at later along the lessons, in which we shall have to see how they participate in making us one who we are, the features we possess, the proteins we do have, not the ones we eat from food, not fish, not beef, not what, but the ones the cells are able to manufacture themselves, but definitely using what we have given the cells as nutrients. So, now the question is, when we talk of accord, what exactly do we mean when we say accord? I hear coding, those who do ICT and stuff like that. But then we are connecting this into genetics. So what's the connection? That we look at this as the genetic code, which is actually our lesson today. When we say something is coded, could it be hidden information? Could they be letters or figures or like the way you write your passwords? How many of you have bought airtime? When you ask for meet you and stuff like that, what are the figures on the card? How do you feed them in? So in today's session, as the background picture shows us that we are having our DNA clearly seen as a double strand, and then out of nowhere, something stretches out as a single strand, and we have words like triplets. They are not twins, but triplets. Can it be a 3-letter section? But in between this DNA and the proteins, what is there in between? What's the connection? What brings this appear here? Which takes us back to things concerning our very biology right away from the beginning. That we always looked at parts of the cell and their functions, and one that is always basic and everyone knows, even pure one, they say the nucleus in the cell is the command center. It does everything. But how? What's the connection between what is inside the nucleus and our muscles and what our body does? That's what we want to look at today. The topic is a little bit complex and rather long, so we are splitting it into short, small, simple lessons that you're able to track bit by bit. So today is only the code and how we can find a connection between what is inside the nucleus and your muscle, and how you behave, and your characteristics, which is here. So there must be something like a bridge. There must be something like a thread that puts our nucleus and its content and links it with what we have as features. So what do we have here? We are all aware that each cell in an organism contains genes. It has all the information to determine who we are. But where is the store? And in which form is this information stored? Which takes us to bullet number 2. Why do we refer to it as secret, encoded information? So those who do serve in the military, you actually know what I'm talking about. How do you communicate? How does a signal get transferred into an action? So, it is a code because this information has to be interpreted, meaning that without any interpretation, without any breakdown, this information is useless. Which takes us to what exactly this inclusive education has brought up. You'll find that some of these students that have visual impairment, check the words I'm using, have their special machines where they type, and others use some pin-like structures and small boxes, and they keep chopping on a piece of paper. Special, very special. But you cannot mark that piece of work as a teacher unless someone has transcribed it and changed into the usual words we know as teachers as we work. I would refer to that simply a fair representation of what actually happens inside the nucleus of the cell. Get the information, decode it, so remove the locks, expose it, make it available for reading. But the question is, how much should be read and for what purpose? So that we finally can have a structure. The codes we get for cash power, the codes we get for airtime cards that are no more now, you go to me to you or you to me, I don't know, whatever you call it. But we also know that the advancements in science and technology that has gone far ahead to look into the nucleus and check what is there has given us some bit of hope. So much breakthrough going on. How many genetic diseases are known? How do we know that they even exist? If I am sickle cell, or if I have a sickle cell anemia, if I'm a sickle cell patient, that's a wrong type of red blood cell. It's a wrong form. But how does it surface? How is its structure connected to the DNA I have in my cells? They're saying whatever we do have as features, as characteristics, all the information is embedded there. All you see the government going through means that there is a constitution they follow, a book, a collection of all the laws governing a state. So they read word by word. After 5 years, we shall have elections. So for us, we only see elections going on. But there is somewhere it is extracted and then interpreted and then put in action. So having to know that this is within the DNA, and we have been able to have great scientists going miles ahead and being able to identify what actually happens in there, having to make a lot of breakthroughs through genetic engineering, as in get the genes, modify them, transfer them into other organisms, locally made or in a modern way make substances that are within other organisms, and we put them in the lab and we put them in containers and we make these requirements we need. Are we able to tell genetic diseases? And if we know that they do exist, is there a way we can minimize their reoccurrence in the systems? Ever heard of genetic drugs? I'm not very sure. We all know that. But exactly what happens here that we have to get into the nucleus, pick out the information, coded, so decode, like you know the way we have decoders at home. You buy the air card and you put in the digits, and then you have signals, you have the dishes on top of the buildings, and then a signal comes. You have what we call a decoder. What does it decode? It decodes the signal. And then out of nowhere, you see your picture on the screen. Like exactly what you're now watching back home. Your TV sets receive signals. But we must have decoders to get that signal, read it out, interpret it, and transfer it into a picture that you see right now. But how does this actually happen within our body? How do we get our DNA moved to mRNA? What is the link between the DNA and the proteins, us, what we have in our body? What is the bridge that puts them together? So here we have a picture, your DNA, and then you have the mRNA codons. They are pairs. They are triplets, 3-letter codes. And these have to be translated until we have our polypeptide, a collection in chains of amino acids making up a protein the cell makes. Remember, this is not a protein you eat as food. It's what your body makes out of the food you've eaten. If you take fish, if you take meat, and other related proteins. How does the body rearrange these amino acids after digestion, absorbed by the cell, and arranges them into your fingernails, into your hair? Beautiful one. Into all the parts that you do have, into the characteristics, into the way you behave, into how your body reacts, the general metabolism. So every section of the DNA is called a gene, which we now refer to as a cistron. So genes you've known, cistrons now could be a new term. Now, it is that small section that will carry the information. So what we display out on our bodies is actually a representation of what is embedded within our nucleus as information. But what do we know? As in, everyone is expected to know this. The function of the nucleus is to dictate all cell activities. Senior 1, that's the definition you're given. But we at this level now go a bit deep inside and break it down. What exactly is this dictation? What exactly is this command, and how does it flow? High command office, through the seniors and then the juniors, until way down to the patrol on the way, get the thug to prison. Action. Is it exactly what the nucleus does? So, we need to make this section of the DNA as the gene, the cistron, active. Wonderful statement. Activating the cistron. Meaning that while all cells, like at the beginning we said that all cells have the same genetic information all over the body. Every cell you have in your body has the same type of information. But each cell will display a feature because that section of the gene is the only one active, while the others are inactive. Which takes us to specialized cells. The grade 11s, you looked at that in your senior 4. The grade 12s, you looked at that. Even you, the senior 1s, you looked at specialized cells. How does a cell become special that it performs only one duty and cannot do the other? Yet, we just mentioned that the nucleus has all the DNA with all the information. What is the purpose of the other information? What if we just discard it? After all, it's no longer relevant. So, once activated, the information is now transferred from the DNA into a new component of a nucleic acid referred to as the mRNA, which now has codons, triplets of bases, as you can check. Now, there is something I really want you to recall, that while we have the DNA double-stranded, here we have single-stranded. Check the letters. There must be something missing. Did I miss it out, or it's what is expected? Think about it. Then when we have this information that is now activated, transferred into the RNA, which we now call the messenger RNA. So it is, you remember the posters, the letters, then delivery, then you open, then you start acting. This will act as a template where we can now finally have our protein. So essentially, what am I trying to talk about here? How does life flow? If we say biology is the study of life, where does it all begin from? Meaning that if we look at what we just mentioned, that moving from the DNA to RNA, we are transcribing. Remember, we used an example of the blind. Visual impairment. They have their special keys, they have their special pens they use to write, but you cannot decode that message unless you know how to. So these teachers would actually be looked at as mRNAs and the ribosomes, remember them in cell parts and functions. And then what they write is what the teacher in your exams is going to be in position to start marking. You only mark what is transcribed from that candidate. So when you see them using their special typewriters, if I would use the words, they actually are coding, if I use the word. Then the teacher will decode their scripts and then pass them on to REB, and then when it comes to marking time, they are also marked as you. You finally get the score. So we'll therefore say that the score is actually our protein. So, our candidate is here, the teacher is here transcribing, and we finally have the protein, which is the true words in the exam, finally written by the teacher, for the other teacher to be able to mark. Because the other teacher, the ordinary one, like me, I can't read those coded structures unless they are decoded, and then put there for me to be able to see. So, what am I talking about here? The DNA, as you can check, the 3D image is actually telling us the nucleus. Check the pores, think about what is pulling out, and then, as it gets out, what is the design? Why this time round not double-stranded? Can you think about it? It looks like a single one. It looks like a millipede. Jesus Christ. So, where do we head to? Can we be able to remember this structure? Could it be like a basic? A pot? Could it be maybe, if this is the nucleus and this is the cytoplasm, then I would easily say that this is most likely to be what? Ribosome. And then we go. Finally, where are we? Our proteins, our nails, our hair, our pigments in the eyes, and stuff like that. So, what exactly do we have here? When we talk of the central dogma of biology, as the chart makes it so clear here, let's follow it together, that DNA can have replication. So we have constantly replication taking place here. All the time, cells are dividing, we need more copies of the DNA, we need enough so that we have each cell produced having a copy of the DNA. But for what purpose? When these cells are finished dividing, What's their role? What is this information that is coded here that unless we transcribe, extract the data, process it until we get a protein? How is my skin, the fingernails, the hair showing a backward link back to the DNA? The word cytoplasm and the nucleus is a clear indication of where each part takes place. But are we having any possibilities that there could be another direction of life, like reverse? I am very sure that you have in some way, covered about things to do with viruses, retroviruses. Why do we call them retroviruses? Where do we classify human immune virus? Why does it attack our DNA and then come out? So it's like it has a reverse gear, I would say. Back, turn around, and continue. Boom, off we go. So, this simply shows us the connection between the DNA and the protein we make. Bridge is mRNA. The process is transcription from DNA to mRNA, and then we translate the transcribed message in the cytoplasm to form a protein. This simply means from the codes we used to have on these air cards, which are normal to momo, codes, star, hash, pra, whatever. Me to you, others say it should be you to me. Why should it always be me to you? It should be you to me as well. What about cash power? When you buy these codes and you feed them poor and then you don't see the lamp, and then you say, okay, let me feed in the codes again. So when this information or data, the cistron, the other section of the DNA, the gene, is used to make a protein, then we shall say, now, the genes we have been displayed, that we do possess them. So if I have sickle cells, then it means that I have a wrong section of the gene. If I have poor skin pigmentation, then it means that within my section of the gene there is something wrong. That's an expression, the display, the showing. So we shall say, once this DNA finally appears here, then we shall refer to that as gene expression. But something we need to remember. How would we pick out this information and throw it in the cytoplasm? What actually goes out? Like we just said before, 1, that our RNA is single-stranded. 2, carries a U instead of T. And 3, like we said in the very beginning, the sugars that are present. Check DNA deoxyribose sugar. The difference lies here. Check where my laser is and on the ribose sugar where the laser is. That's where the difference is. And then the bases. We already talked about them. A, then U. So are we in position to write out these letters and wrap them up? If we are saying during translation, the DNA acts as a skeleton, as a template, where mRNA can be made. Remember, complementary base pairing. A with T and G with C. But when it comes to the mRNA, what do we have? Kick out the T and replace it with a U, as we do have here. So can you finish that up? Pretty simple. I did the first 5 bases for you. Can you wrap it up? Twaji. But what do we know? It is true, it's a fact, that we all have the same way in which our proteins in the body are made. as long as you're a living organism. The language is the same. The chain of life is the same, from the DNA to the protein. So whether you have bacteria as examples of prokaryotes or us as examples of eukaryotes, we all have the same language that is used to have life moving on. So what does this mean? That when comparisons have been made between the DNA bases and sequences in humans and other animals, there is so much relation that a certain protein made within the bacterial cell is done in the same way in my body. What does that mean? That the production of human insulin in bacteria, as an example of the universality of the genetic code, is one good one we should not just take simple as that. There is so much in it that we still do have useful bacteria. If I'm diabetic, I don't need to depend on an animal to get insulin. So they can genetically engineer bacteria to be able to produce the insulin that I do need. So an identical genetic code is used in both prokaryotes and eukaryotes. Thus, bacteria can be genetically engineered to make human proteins like insulin. If I'm diabetic and someone is not, it's very risky to get his blood, sample it down, filter it, pick out insulin, and then give it to me. What if I have an infection? What is the ethics behind it? What about religion? What about the chances for infection? So can we go bacteria? Does it have rights? Can we take advantage of it? Monitor and manipulate its genes and have the simplest structure of insulin, pack it, give it to me, off I go. And we can have large amounts, since afterward bacteria produces much faster. And what else do we have? That all living organisms, it is ATUGC. They are the same. So the code is similar. This image you see on the screen is about how actually forms of DNA, cistrons, can be moved until we insert them in bacteria and then give them a chance to have the proteins we do not have. So the sequence remains unchanged, and the proteins would actually basically look the same. And finally, where did we come from? Look at the tree of life in terms of genes. Who had the first code? Who borrowed the code from the other? How has this been transferred? Conflict now, the controversies. Where do we place the Bible? Where do we place science? And how can we intermarry the two so that we do not have a lot of controversies? So with that, I would say, take it and think about where you came from those three categories of life. Bye.